So we have Belinda, Dr. Belinda Hartmann is the director for IP at the University of Queensland, the commercialization entity. Belinda's IP portfolio encompasses all registered IP rights filed by the University of Queensland, which is primarily made up of patents and patent applications. Her academic history is intrinsically linked to the UQ and the St. Lucia campus in Australia, having completed her PhD here at the Bioengineering and Nanotechnology at the ARBN, before taking on a postdoctoral fellowship at CSIRO in the QBP. Since leaving the lab bench, Belinda has qualified as an Australian and New Zealand patent attorney, working in some of Australia's largest IP firms, with a specialization in the drafting, prosecution, and enforcement of life science interventions with a focus on the pharma sector. With that, it's a great pleasure to welcome you, Belinda, and thank you so much for joining us tonight.
Thank you, Sandra. So now let me share. Okay, I'm going to work on the assumption that we can see that.
Yes, we can see it.
Okay, so thank you for that introduction, Sandra. And yes, we're going to talk about some lessons that I've learnt the hard way through my experience either in private practice or now in a research environment at Uniquest, which obviously is linked to UQ. I'm going to preface this by saying that I'm not a lawyer, I'm a patent attorney, so I do have a law degree, but purely in IP. I can't help you with the parking ticket, and so some of the civil legal questions may not be right up my alley. So I'm going to also start with an acknowledgement of country. From where I'm sitting in Brisbane in Queensland, Australia, we acknowledge the Turrbal and Yagara people, which are the traditional owners of the land where my flat is. And we also acknowledge, being in the innovation sector, that the traditional owners in Australia were actually the first innovators for our nation. So in the next 20 minutes, and I hope it's 20 minutes, I'm going to cover off 4 topics where IP issues can arise. And the first one we're going to touch on is the IP encumbrances, which typically have their foundations before there's even IP. So we'll look at how to avoid some of those traps. Speaking of traps, we have to talk about the disclosure traps that people fall into when looking to file registered rights for IP. And then part of that is also finding the right time to file a patent application. And lastly, whilst I'm not an expert, I am going to talk a little bit about AI in an IP or sensitive or confidential environment and some sort of key considerations we need there. I promise, and I hope to hold to this promise, that there's not going to be any case law in this presentation. Yes, I am an attorney, but I'm going to try and make sure that this is more of a practical lesson as opposed to case law, which, let's face it, is pretty boring. Okay, so let's talk about IP encumbrances. So this is where, in my world, upstream decisions that are made without my knowledge or without many people's knowledge actually can have significant impacts on downstream opportunities. And when I say opportunities, I mean opportunities to commercialise useful and exciting intellectual property. An IP encumbrance is really just anything that's going to put up a barrier to that commercialisation. It's a limitation of the rights and what you can do with those rights. It's never going to, well, not necessarily never, but it may not be an insurmountable barrier, but it just makes life a little bit harder. In my world, IP encumbrances typically occur when you're getting money or materials or help. So things like grants, research collaboration, purchase and material transfer agreements, they're all going to have IP clauses in them, typically, because there's a, they want to sort of put a flag in the sand as to what's going to happen in that IP. And overall, if we talk about the mistake that people are going to make with IP encumbrances, is that there's an assumption that nothing is going to come out of these agreements. But I think best practice really is to assume that there will be a commercially relevant outcome out of any agreement, and based on the IP clauses in that agreement, consider the fate of the IP. So let's dig down in a little bit more detail for MTAs. MTAs are routinely a source of great stress and anxiety for me because the volume of them that go through universities is massive. They're typically deemed, innocent and innocuous. They get executed quickly, but they are a source of an IP encumbrance downstream quite often. So an MTA is a material transfer agreement. It basically sets out the grounds within which you're exchanging materials or information between a recipient, which would be you, and the provider, which is the entity with the material. The agreements typically aren't that extensive. They're not 50-page documents. There might be a few pages. There'll be a selection of clauses, but you will always see clauses to the effect of the terms of use of the material and distribution restrictions. And you'll see on the slide, I've just put up an example. I've just extracted some excerpts from a really starting point template for Uniquest material transfer agreements where it's outward facing. And this is the starting point. So they're actually quite tough, these MTAs. But you'll see that this clause, it talks about what you can do with material. It says, you can't use it for commercial purposes. There's the typical caveats of not for human use. And it also talks about, controlling the material, because the material transfer agreement is also about the security of the material, so it's not getting out in the public domain. We'll also see confidentiality restrictions or obligations. These obligations to disclose breach disclosures will come up more in MTAs, because there is an acceptance that mistakes happen and there are breaches, and so there is an onus on the recipient to disclose that. Also, importantly, we're going to have IP clauses. So you can see for this particular MTA that the IP clauses are pretty tight. The material and all the derivatives are owned by the provider. The new IP is owned by the provider. And so, that it's tight for this particular MTA. But once again, this might be an MTA we'd use for really valuable IP or as a starting point to negotiate that material transfer. So that's all well and good. That's a theoretical example, but let's go on to a real world example that came across my desk recently. And it's an MTA for a plasmid, a useful plasmid that was provided from a non-profit organisation that has a repository of biological agents and reagents, including a vast library of plasmids. So if we're thinking about how upstream decisions have those downstream implications, here were the upstream decisions. And, it's not the end of the world. This thing happens, but this is actually how it has happened in this instance. So the researchers needed the plasmid to conduct their research, and the only place that had the plasmid were the providers. So, they had to execute an MTA, which they did, accepted the material. They went off and did what they do best. They did great research. And in that, they actually happened to develop a commercially relevant innovation. Unfortunately, well, that's not unfortunately, it's just a statement of fact. The innovation is actually a modified plasmid that relies on the material plasmid. It actually relies on the backbone of that plasmid and some other features of that plasmid. So we know now that there's the commercial potential in this modified plasmid. So we now have to go back and check the MTA. Ideally, you would have been aware of the restrictions in the MTA before doing all this, but this is actually typically how we find out about it. So I'm actually going to use excerpts that are anonymized from this particular MTA, so we can actually go through it together. So for use and distribution, we're looking at that. It says, okay, teaching and research purposes. That's really all they've done so far. And there is restrictions on whether or not you can transfer the material around. So fairly standard, nothing too stressful yet. And then when it talks about the material, it sort of indicates that modifications are outside of the scope of that material. So this is great because we have modified the material, and it gives the indication that modifications of the material are allowed. It's not excluded under the use provisions. So all fine so far. Also, if you look at this B clause, you'll see that there are, you can actually distribute this plasmid to other non-profit organisations for research and teaching purposes only. There is another caveat below that you need permission. But if we look at the C clause there, so whilst it sounds negative because it says without written consent from the provider, we can't commercialise, but the fact is it indicates you can commercialise with written consent. So far, we're not, that there seems to be a workaround here. The problem arises, so the implications so far, research only, we can modify the material, we can distribute, but we do need explicit permission from the provider to do distribution beyond non-profit, which inherently would be required for any commercialisation anyway, and also for the explicit process of commercialisation. So when we get to IP ownership, things get a little bit tougher, because whilst the beginning of the sentence sounds great, the recipient retains ownership of modifications, the caveats are quite clear there that if the modifications actually contain the original material, then we might have an ownership issue. And so this is something that needs to be investigated, but there's not a clear line in the sand between our modified plasmid and ownership by the provider. So we may be sharing ownership there. We also see this clause that when it comes to actually going down the commercial pathway, that the provider needs to be involved. The provider, besides the fact that they need to provide consent, there may also be the opportunity in providing that consent for them to put conditions in that, and those conditions might actually be related to being involved in that process. So the downstream implications now are, okay, we still can potentially commercialise. The provider may be a co-owner of the IP, but we'll actually have to investigate that if the modified plasmid still falls within the scope of containing the original material, and that will require some investigation. The provider may end up being a party to the commercial process if they give consent and they choose to be. And then if it's actually successful, the provider, depending on the commercial arrangement, may actually be entitled to royalties. So not terrible. It's workable, provided we know about the MTA. Notwithstanding that, there's no commercial opportunity with respect to this modified plasmid without the provider. So, you know, when we think about MTAs, sometimes you go so far down a pathway that perhaps if you knew about these conditions, if you actually looked at the MTA before executing, you might have actually looked for an alternative than getting the plasmid from this particular source, or you may have built your own. So that's kind of the downstream implications of this way that's actually panning out. Let's look at a less responsible way that this could pan out. So we go through all the processes. The innovation is the modified plasmid using the material as a backbone. But this time, you either ignore or forget about the MTA. So for all intents and purposes, no one asks, no one knows, and no one remembers that there was an MTA involved. So what happens from here? Here's some hypothetical scenarios. So you've got this great modified plasmid, it constitutes a new invention, so you file a patent application. But you haven't listed the provider as a co-owner, despite the fact that we've established that the provider may be entitled to ownership of this modified plasmid. So now you've got a validity issue of your patent application, because a patent application or a patent has to correctly list all the people entitled to that invention as owners. And not having that list correct is a ground for revocation in a lot of jurisdictions of a patent, because you're not entitled to it. A next step might be that you go to a commercial animal trials facility with the modified plasmid and also the control plasmid as the material for testing in various experiments. So now you've actually breached the conditions of the MTA in terms of distribution, because a commercial animal trial facility is not a not-for-profit. Then, if you're really lucky, you may enter into a commercial agreement to further develop that IP. So inherently, you've breached the distribution, because to commercialise, you have to put the material out there. But in that commercial agreement, you've probably made warranties to the other pharma company that you're entitled to this IP. There's no other legal encumbrances. There's no barriers to this commercialisation. So you've made warranties that you can't stand behind. And also the provider was meant to be at least consent to that commercial pathway and may have wanted to be a party to that. So this is not a good scenario. And really, no one cares about these things typically until things start making money. But if this was a success, then your success is built on a sandcastle, really, because your foundation IP is potentially invalid. You've got vulnerable commercial agreements, and then you've got the threat of litigation, potentially from the provider, or termination of your commercial agreements from the pharma company. So this is just and this definitely can get quite close to happening. So in terms of the agreements, just always look for those IP clauses. So that's lesson one for today. Next lesson, let's go. So you want a patent. This is a bit tongue-in-cheek because dealing with a lot of researchers, there is a lot of like, hey, give me a patent. I want a patent for this invention, which is kind of amusing to me because you don't just get a patent. You file a patent application and then it has to go through examination in every jurisdiction that you want that patent in before you can actually get that granted enforceable patent right. And there's various stages of this process, but two critical thresholds that you need to get through to go from patent application to a patent is that the invention must be novel and inventive, and that's more about actually the invention. But then the patent application needs to have the appropriate support, enablement, and sufficiency, which is more about the actual document that you file to support that invention. So if we're thinking about novelty and inventive step, we should all probably be familiar with these concepts. But an important thing to note is that your invention needs to be novel and inventive in the context of the prior art, and the prior art is any information in the public domain. There's no geographical restriction, no language restrictions, and patent officers, they're the ones that do the examinations. They don't care how the prior art ended up there. So if you put prior art in the public domain about your own invention, the patent office doesn't care. There's some grace period provisions, but you should assume the patent office doesn't care that you actually did it. So when I'm having conversations with researchers about their proposed invention and I ask the question, so have you publicly disclosed this? What I'm really asking is, have you added to the prior art in a way that compromises the novelty of your invention? And unfortunately, the answer is sometimes yes. So we're going to have to go through this. I know that everyone knows a lot of this, but we're going to have to go through how these examples of how you can put your own prior art out into the public domain by your own self-disclosure. So scientific articles, we know all that happens all the time. But you know when you're doing your submission, is that confidential? Is the review process confidential? If you're trying to time the filing of a patent application to just before publication, are you going to get advance notice of publication? Do you know if it's going online before the actual journal is released? These are questions that you need to know the answers for. Oral and poster presentations, this has happened actually a couple of times just in my few years at Uniquest. We time a patent application related to when someone's going to give an oral presentation, but unbeknownst to us, the abstracts are being published in advance of a conference. Another thing that actually an incident came up recently is that there was a lot of research institutes will have internal closed poster and abstract sessions, and so that's sort of a confidential environment. But it ceases being confidential if people take photos and put them on LinkedIn. So just be aware of that. Just for grant applications, just be aware, because grant applications are typically really detailed and they have all your current IP plus all your future proposed IP. Just be aware if they're actually going to be published if they're successful, or if milestone reports are getting published during the grant life cycle. Clinical trials, so this is out of your control. Clinical trials, the protocols and results need to be published by government organisations in most jurisdictions, but you should have an idea, you should know when that's going to happen. One thing that people don't always anticipate is that things like patient consent forms is those stages well before the clinical trial commences. If there's enough information in there, that can be a prior art disclosure. Theses, don't forget that when you submit a thesis and it gets approved by university, your university chucks it in the library. You can embargo a thesis with your library, with your university, but also note that it will typically have an expiry date. So if you're trying to time the filing of an IP, a patent application, you want to make sure that your thesis is still under embargo until that happens. And lastly, news and social media. I'm going to go through an example of this because I get it. We love media attention for research activities because it generates excitement and interest and furthers commercial activities. You've just got to be careful because I think people, simple media releases that might just be a couple of paragraphs don't seem that substantial, but it can give the game away. And on that note, let's just have a look at a little example. So I should say that this media release wasn't problematic at all. This was all fine. It's just one that I found that was really easy to demonstrate my point. So simple media release, this is an excerpt. When I go through the core features of this media release, and I'm thinking about it in the potential of it being discovered later as a prior art disclosure for this particular research activity, I'm looking at the key features. And as a patent attorney, I can take those key features and basically I know what claim 1 would be for this invention. I know that it's specifically to glioblastoma. It's this particular chemotherapeutic agent. It's porous silicon nanoparticles, and the advantage is that it crosses the blood-brain barrier. So that media release, albeit a couple of paragraphs, would be novelty destroying for an invention based on this media release. But it didn't have to be. Really simple amendments could have de-risked this media release while still putting the message out there and still actually getting publicity. So take away the particular drug, take away the particular cancer. I took away details of the silicon nanoparticles, in part because UQ has got a really renowned research group that does a lot of work in silicon nanoparticles. So if you Google that, you would have gotten a really detailed description of these particular particles. But it is just an example that, people like myself, we don't want to be a barrier to getting information out in the public domain, but it just needs to be done with some consideration for what might be a valuable future IP position. So just quickly on the support and enablement, this is a difficult discussion sometimes we have with researchers because they've got a great idea and they want to file really quickly. But because I said that the support enablement threshold and the novelty and inventiveness threshold, they're weighted equally. So you can have the most incredible invention, but if you don't have enough detail in the patent specification to provide enough information to reproduce that invention, you won't get through patentability. You won't get a granted enforceable right. So what I deal with a lot is trying to find this balance between filing a patent application too early or too late. So too early, it's a great idea, but just an idea. We always want the researchers to come to us early, but we can't pursue those rights that early. We need that proof of concept. And the threshold for proof of concept for a patent application isn't as high as for a study or a trial or approval or even a paper. It really is to demonstrate to the Patent Office, and they use the term that you're in possession of the invention. Have you made the invention? Does it work? Down the other track is too late, and that might be your own disclosure, or you may just get scooped. So, in really crowded IP spaces, there's quite a lot of tension between too early and too late because you want to have that support and enablement, but you need to find that sweet spot in the middle. And so, filing a patent application is a balancing act of timing. But enough about that. I'm running out of time, so I'm going to go through some AI stuff quite quickly. So AI, great, love AI. Not an expert in AI, but I think if you're working in an industry that has sensitive information, so confidential intellectual property, trade secrets, you've got to be careful about AI. So the first message I would say with AI is that it's bad at IP law, and I'm not saying that to protect my profession, but AI just doesn't do well with this intersection of science and law, and claim language, if you've ever read a patent specification, it's just not a normal way of speaking or communicating. And every year, a rather large law firm in Australia does a controlled study of all the presently available AI tools, giving them a specific set of legal questions from different fields with increasing complexity. And over the years, they have demonstrated that, yes, AI is getting better for law in general, and for really basic legal questions, they found that the best AI tools might be as good as a junior, albeit mediocre lawyer. But in terms of fields of law, AI is bad at IP. It consistently performs really badly in IP. So anyway, the message there is don't use AI for IP advice that you're not capable of confirming yourself properly. The results look very convincing, but for now, it's quite unreliable. Secondly, AI is a terrible gossip. So the information, if this is open source AI tools, remember that this is machine learning, it's training. And so your confidential information that you could be putting into an AI tool could eventually be an AI output, which could constitute a disclosure. It can be a prior art disclosure. And in the IP world, it's something that's making us all very nervous. So if you are going to use AI tools, be really careful. Review the confidentiality policies of any tools you're using. Preferably just use closed loop tools. And another thing is, and this will come up in the next section as well, is you really, I would say in general, if people are using AI for any work-related purpose, start documenting the prompts that you're using, because if it comes out that, you know, your information is, your confidential information is somehow being spat out by an AI tool in response to a similar prompt, and this might work against you as prior art, it's good to be able to document the fact that, you know, it was a self-disclosure. There might be grace period provisions, but anyway, document your prompts. Another reason to document your prompts is, so AI cannot be an inventor of a pattern. This has been a massive case around the world where it's been challenged that for a particular invention that AI should be the inventor, but it's unanimously been decided that a patent inventor can only be a human, but a patent application must list all valid inventors, which gets complicated because AI can actually be inventive. So there's a potential issue for the validity of a patent application if you've used AI tools that have ended up contributing to the core inventive concept for that invention, but you can't list them as an inventor. So therefore, is the owner fully entitled to that invention, or is it possible that the AI company could end up being a co-owner of that IP? Is that where the contribution is going to be? So once again, just be really open about any AI usage that your AI usage in these fields. And this is probably where it's more important to be documenting your prompts, because if down the track, you know, the legal system takes a while to keep up with technology, if down the track there's a question mark over where the invention came from, and if AI was a contributor to that invention, having your prompts documented can demonstrate that no, the AI was just responding to your inventive query, or that you were actually, AI was just doing what you told to do it based on your inventive contribution. But yeah, once again, not an expert in AI, but just be careful. So in summary, oh good, Sandra, I'm so sorry. So I think when you're in an area where there's the potential for IP, just assume there will always be IP and act accordingly. Don't ignore the limitations. Don't pretend that MTA doesn't exist or the IP clauses of your grant doesn't exist. You can work with them or work around them as long as you know they're there. Don't inadvertently get yourself in more trouble than you ever needed to be. I think we'll see with a lot of the sort of discussion points today is timing is everything, and it can be a bit of a balancing act, but knowing the timing of things is really important. So don't pretend that, you know, everything's just going to fall in place. Managing disclosures is really important. Disclosures aren't per se bad. You just need to know when they're going to be and make sure that, for example, if you're filing a patent application, it happens before your disclosure. And lastly, just use AI cautiously. We don't know how it's going to play out in the IP world at this stage. So yeah, that is probably going to be it for me, Sandra. If we still have time for questions, and we may come back at the end. I don't know how we're going.
Hi, Belinda. No, thank you so much. It's been absolutely amazing to hear all about that. Now, for all the attendees, so there's a Q&A session that you can just post in your questions. I would have two burning questions, if you allow me, Belinda. So one of the questions is, does every university have a legal department like what you have here at the University of Queensland with Uniquest? Does every single university have that? Or is it Australia-wide, worldwide? Is it everywhere the same?
So, yeah, I mean, every university has a legal department. It's just, I suppose, at the University of Queensland, because Uniquest sits outside the university, even though we're wholly owned by the university, there's kind of a disconnect between the upstream legal and the downstream legal. So at Uniquest, we're all about the IP commercialization. So we're about, you know, at the point where you have something commercially relevant, and then we're the ones that are going to run to it. But all the legal stuff that takes the researchers to get to that point. So you know, UQ Legal, like there's massive legal department and they manage all the grants and the research agreements and all the MTAs and all the CDAs related to that. And so, but there's a huge volume. So the fact that the fact that the MTAs cause us grief downstream, it's not a reflection of UQ Legal. It's purely like just the volume that they have to process. They can't be reviewing these documents all the time. And for my part, I kind of think that maybe the researchers need to be looking at the MTAs a little bit with the lens of what's going to happen or what are they actually going to do with it, because UQ Legal doesn't know what they're planning on doing with the material. And then in Uniquest, so that's, you know, our TTO, we have a decent in-house legal team. So there's 2 lawyers, we have a contracts specialist, I've got an IP assistant, and we've got an in-house patent attorney, me. And that's a volume thing. That's because of the size. Not all universities would have that within their TTO. That outsource quite a bit of that. So we're very lucky.
Amazing. Now, one last super quick question is, researchers are normally using lab books, right? So how much detail, how much emphasis is put on, number one, recording the information in the lab books in paper, or is it sufficient to have an electronic record that is not signed and not dated? What is the take on there at the moment?
Yeah, so that's really interesting, because that was more of a concern sort of 10, 15 years ago when the US was still a first to invent jurisdiction, whereas now all the jurisdictions in the world are first to file. So the patent offices don't actually, for their intents and purposes, your invention doesn't exist until you file the patent application. So gone are the days when we had to go back through lab books to find out the date that an invention occurred. What we do use lab books for, and where it comes more into play for us, is determination of who's the inventor, so the inventorship who contributed to the core inventive concept. But the lab books versus electronic lab books is going to be a policy issue. And I know at UQ there's institutes that use digital lab books. It's not enough to just have like Word documents, because digital lab books actually have like version control and timestamps. But the digital versus paper, it makes no difference as long as there is the appropriate documentation. So I think a lot of it comes down to the timestamps, and, you know, because you can see even a digital lab book, you can see who's making each contribution, like whoever is in that lab book making that contribution.
No, that's great. Well, thank you so much, Belinda. With that, I would love to welcome then Andrew, and then we have at the end time for additional questions for both of you, if that's okay with you.
Okay. Yeah.
Great. Thank you so much. Now with this, I would love to introduce Andrew. Andrew, you completed your PhD at the University of Queensland in 2019, where you worked on generating stabilized subunit vaccine candidates and the proof of concept diagnostics for Ebola virus and other related filoviruses. His PhD supervisors were Paul Young, Keith Chappell, and Daniel Watterson, the 3 co-inventors of the molecular clamp technology developed at UQ, and that recently got sold or acquired by Sanofi. Since 2019, he has performed contract research for the spin-out company that uses the molecular clamp technology to make vaccines for common respiratory pathogens. That work has resulted in a bivalent RSV and HMPV, the human metapneumovirus, vaccine that is currently in a phase 1 clinical trial, with a first in human phase 1 clinical trial of a trivalent vaccine targeting RSV, HMPV, and PIV planned for 2026. So with that, over to you, Andrew. Look forward to your talk. Thank you.
Great. Thanks, Sandra. Just trying to get my slides up. Let me know if they're visible.
You can see the slides, but not in the presenter view. We still see the slides on the left. Presenter view. Great. Thanks.
All good? Great.
Yeah.
All right. Thanks, Sandra, and thanks, Belinda, for a very important talk before. So I'll just crack on with this talk. A bit of a change of pace. Going to talk about, I guess, viral class 1 fusion proteins for a while, and mostly just sort of describe what they are, how they function, why they make good targets for effective vaccines, and how we can sort of apply our sort of deeper mechanistic understanding of these antigens to rationally kind of design more potent, more effective vaccines. So these are antigens that are presented on the surface of a number of very clinically significant viruses. In the pictures here is kind of a subset of some of the antigens we've made with the molecular clamp. These are not to scale. These are just like representations. But what these proteins all have in common is they're these 3 repeated subunits that kind of coalesce into what we call a trimer. They're presented on the surface of the cells. Typically, these viruses are enveloped viruses, so they sort of bud out of the host cell. So immunogenically, they kind of look like self, except for having these spiky bits kind of poke through. Other viruses will have, a secondary antigen or multiple antigens that do poke through the membrane, but often it is just this sort of fusion protein. And one of the reasons why, or a couple of the reasons why they make great targets is they're an available target, they're presented, and they also have this critical functional role in cell entry. So if you can sort of suppress that in some way, you disrupt the replication. Now, everyone knows spike. This is the one that everyone's aware of now. That's a class 1 viral fusion protein. As Sandra mentioned, my PhD was studying the Ebola antigen, as well as Sudan and Marburg. And then since finishing my PhD, I've been focusing on these 3, which are just really common respiratory disease antigens. So let's zoom in a little bit. This is all Ebola virus, just as an example. Up here, marked with A, I'm not sure if you can see my cursor. Hopefully you can. We can see sort of the protein schematic of how these proteins look when they come off the ribosome. So you got this kind of this GP1 subunit. The nomenclature for these is not consistent. So for Ebola, it's GP1 and GP2, which is glycoprotein. For spike, it's S1, S2. For hemagglutinin, it's HA, and so on. So in the Ebola context, we've got GP1 and GP2. So GP1 is kind of like the more outer solvent-exposed subdomain. There's then cleavage by a host furin. And then the GP2 subunit in this case kind of is the mechanism. This is the thing that drives the actual fusion of the host and the viral membranes that allow the virus into the cell. So this is, if we think about like how a virus actually enters into a cell, there's kind of these 2 points of entry. So you've got this initial attachment and entry where you just kind of bind to the target cell. This is often, well, this is kind of mediated mainly by these GP1 units. So we say that it modulates tropism. So if it's got a kind of a generic attachment profile, so it can attach to a variety of different cell types, that gives it a broad, I guess, range of targets of cell types that it can then infect. And that's kind of mediated by this GP1 unit. So that's the first bit of internalization. In the case of the GP1 here, what I've circled in around the B there is mostly just space. I just want to mention that these are electron constructions. So what we can't see on these images is basically anything that's flexible, anything that moves around. So what actually extends into this sort of unresolved area is this really densely glycosylated sort of just branches and branches of carbohydrates that actually account for more than 50% of the weight of this whole antigen. So we can only sort of see like a subset of the cap and the core, but there's all this other stuff, and that's what actually interacts with this cellular receptor. So the cell essentially says, great, we got some carbohydrates, we got some protein, let's have that. It starts to endocytose them. So you get this internalization. Then you get the maturation of the endosome. So it starts to become a pretty hostile place in there. You've got just the pumping of protons into the vesicle. You've got proteases that are activated by the protons. In the case of the Ebola virus, these cathepsins kind of activate as this endosome matures, and it results in the cleavage of the majority of the GP1 subunit. So basically 85% of the weight of the GP1 unit gets cleaved off and gets jettisoned. And that reveals this kind of receptor binding site, which is revealed after that cleavage. And then we get to the second stage of entry, which is you're in a vesicle, you need to get into the cytoplasm. And this is what we mean when we talk about fusion. So fusion is essentially fusing the host and the viral membranes together to create a fusion pore, which then allows you to deposit your replication complex for replication. So this is all, as I've mentioned, mediated by the second subunit. So the second subunit is characterized by this transmembrane domain, so that anchors the protein onto the virus itself. It has heptad repeats, so 1 and 2. These are essentially like long alpha helices that can they have a hinge, in a sense, in between them. And then you have some sort of like fusion peptide. So this is a hydrophobic domain. This is what is exposed once you have that sort of that processing in the endosome, and that is what embeds into the host lipid bilayer. So the hydrophobic fusion peptide, embeds into that host bilayer. This is kind of a schematic representation of what might be going on in this, but with our imagery, we can see basically the first phase and the last phase, and everything in the middle here is just kind of theoretical, but it's pretty sensible and probably correct. So what to take from that is essentially that you need multiple of these antigens working in concert to make these pores. If you look over to the right, so I guess a virus is basically just an inert ball of chemicals in a sense. It can't really do much by itself until it sort of drifts around and then bumps into one of these cells. And then it seems to be doing this quite active process of fusing the membrane. And so the question is, how does a virus that's basically an inert ball of chemicals, how does it do anything active? And the answer to that is that it has this kind of potential energy locked up in these pre-fusion conformations. So I like to think of these fusion proteins or these spikes as being in a sort of spring-loaded sort of position when they're in the pre-fusion conformation. So they're relaxed, but they could be more relaxed. And then there's the potential energy that's wrapped up in this pre-fusion conformation that drives the fusion of these membranes as it transitions irreversibly to that post-fusion conformation. And that's the stuff that does the work here, that actually opens up these pores. And so this presents a bit of a challenge for vaccinologists, because the epitopes that are on that pre-fusion conformation are far more neutralizing. They're a much better target than the post-fusion conformation, which makes intuitive sense. You want to kind of put a spanner in the works of the mechanism before it's done its job. But if you then try and, say, solubilize these pre-fusion antigens to try and make them just a vaccine, like a subunit vaccine, often what will happen is it'll just trigger, it'll spontaneously go to post-fusion, and you'll have a weakly neutralizing vaccine. So people come up with various ways to try and stabilize these pre-fusion conformations. Often it involves sort of non-natural disulfide bonds that kind of lock some of these helices, say, that are in close proximity in the pre-fusion, but in more distant proximity in the post. And by doing these sorts of strategies, you can sort of stabilize this pre-fusion conformation. Now I'll introduce the molecular clamp, which is conceptually a very sort of simple solution to some of this problem. So when we make a soluble antigen, we have to remove this membrane anchoring domain because it's hydrophobic. If we left that on, all the hydrophobic parts would sort of clump together and you'd end up with a big aggregated mess. So the molecular clamp is essentially a soluble stand-in for basically the stabilizing forces of the transmembrane domain. And so what does this look like in the lab or in practice? We basically just design expression vectors with the clamp appended at the 3' end. We can then express that in a mammalian expression system. We use CHOs. And then we can purify the antigen out with high purity using a clamp-specific resin, is what we typically use. We've also got MABs that can do it. And so this allows us to basically generate vaccine candidates without having any target-specific reagents, which is great. It has implications for things like novel emerging diseases and rapid responses and that kind of thing. Now, as this is a class 1 fusion-specific technology, it can't be applied to all viruses, but this is the list of WHO priority pathogens, and the majority of them are amenable to producing a clamp-stabilized vaccine. And now I'll move on to what I've been working on more recently. How am I doing time? Okay. So RSV, MPV, and PIV are all just all very ubiquitous viruses that basically we're all infected with, usually multiple times by the age of 5. So we've all got basically a memory immune response to these guys. And if you add up the disease burden in terms of either hospitalizations or deaths, you get quite a similar number when you add these three together to seasonal flu. Now, I've just showed the data here for the US, like over 65 population, but because data on PIV3 is a bit spotty, but this trend does kind of extrapolate out to other populations. And yeah, and if you can develop a good vaccine to these three, odds are you're not going to have to do a seasonal update as you do with flu, because these antigens are far more genetically stable. They don't do the sort of the class switching that HAs tend to do. And they're also, I guess, genetically far more stable as well. They're under less evolutionary pressure. So looking at what this looks like in practice, this again is just one of those schematics of the gene, your F1, F2, and your transmembrane anchoring. So that's what the wild type gene kind of looks like. So we make a negative control. We say, okay, what does this look like when we just remove the transmembrane domain? Do we need to do anything else? And so this is size exclusion data. So going from left to right, we've got high molecular weight aggregates. The blue is what we'd expect the size to be if it was the trimer, if it was the nice 3 pieces together in solution. And then to the right is smaller stuff. So either monomers or maybe degradation products. So this is an interesting representation of these 3 viruses. When you do this, you get quite different profiles. So RSV is just fully aggregated. MPV, you've got a bit of monomer, you've got a bit of aggregation, no trimer. And PIV, you see, actually, there is some trimer there. There is some of the desired product, even when you just remove that transmembrane. Going back to Ebola quickly, Ebola is actually really, really good in this test. It's really nice homogeneous trimer, because one of the Ebola virulence factors is it actually, I'm going to say intentionally, I don't mean intentionally, but some of the presented antigen is actually removed by host proteases and then seeped out into the solvent. So it's kind of like an immune evasion strategy that Ebola does. So it's actually evolved to have a stable trimer without those transmembrane anchoring domains. Okay, so that's the negative control, let's say. We also like to use a positive control. So we use the best-in-class comparators from the literature that use these stabilization strategies that I mentioned briefly earlier. So introducing non-natural disulfide bonds and so on. These also tend to use either a fold-on or a GCN4 trimerization domain. And you can see, yes, you get nice improvements in trimer. And these are all, determined to be good pre-fusion antigens. And then our lead kind of uses a combination of the strategies where we do include some non-natural mutations, some non-natural disulfide bonds and so on, and the molecular clamp. And we get this really nice homogeneous product. And a great thing about it being so homogeneous is there's less loss in downstream processing. So you can maximize your yield a bit more. Regulators like a homogeneous product. It's makes the regulatory steps a bit easier. And we've also added some sort of non-natural glycans to the sort of outward facing solvent exposed parts of the clamp, just to kind of dampen the immunity, kind of mask it a little bit, sort of similar to what the Ebola antigen does to mask its own antigen. When we look at binding with prefusion specific monoclonal antibodies, we see nice binding consistent with that more sort of globular prefusion form as opposed to the elongated, post-fusion form. On the right here, these are micrographs of our actual images of our actual antigen, and you can see the clamp mark there. Conspicuous in its absence is the MPV antigen, and that's because that antigen seems to be far more flexible. It seems to breathe quite a lot, as in these 3 monomeric pieces have much more degrees of freedom. And there are, in fact, some highly neutralizing monoclonal antibodies that bind onto that sort of inner trimer interface of the antigen. So yeah, that's been a harder one to get good resolution on. Looking at yields, we get really nice yields with clamp stabilized antigens. In the purple, those are 50-liter bioreactor scale with stably selected clones. So getting towards sort of 1.5, 4.4 grams per liter makes cost of production incredibly cheap. Stability of these antigens is really great as well. So out to sort of 9 months in the fridge. And this is in a pre-filled syringe. So it's really helpful for clinicians if you can have that liquid stability, because it saves them from having to, mix up on the spot, which is a really enormous sort of quality of life improvement for clinicians. And also, this is just kind of trying to break the antigens by using pretty physiologically irrelevant temperatures, but just trying to see how they perform. So VXB213 is our clamp stabilized RSV antigen. Arexvy is the clinically approved GSK one, and Brisvo is Pfizer's. We've done a series of mouse immunizations. This is looking at basically the reactivity to the tag domain. So whether it's the fold on for the controls or the clamp itself, we see proportionally quite low reactivity to the clamp. Neutralization is alongside DSCAV, which is that GSK antigen. And DSK is E2, which is not in an approved vaccine yet. Yep, so nice high newts. VXB241, I should mention, is the bivalent. So that's the RSV and the MPV together. Here we've just got a couple of extra doses, dose ranging with Arexvy there. And we've got, again, MPV. And this is the PIV newts here. So really nice, strong, potent neutralization. And when you compare to these SOL, so these are the sort of the unstabilized ones, they're not in every comparison. Yeah, down here is SOL. So this is just removing the transmembrane domain, fully unstabilized. Consistent with all the theory of the pre-fusion conformation, you do get that large jump up when you stabilize those epitopes. So yeah, that's what I was to talk about today. I want to just quickly thank obviously the inventors of the clamp, Paul, Dan, and Keith, and the team. It's been a big team. Put in a lot of work, and yeah, it was very rewarding to see Sanofi take so much interest. And because I understand this is an international kind of talk, hopefully there's some Europeans listening. I'm going to be in Portugal next week, so if you're going to ESCMID, say good day. And this is the paper we released in July, that if you wanted to get deeper into the sort of MPV and RSV, just antigen discovery work, yeah, we haven't published anything on the actual ongoing clinical trials yet. But yes, thank you very much.
Thank you so much, Andrew. That's been really amazing. And I think it just shows really how much work it has gone into all of that work altogether. One quick question. You had mentioned before with the class 1, have there been any proteins, any vaccine candidates that haven't worked with the clamp technology from the ones that you've tested so far?
That haven't worked. Well, it depends on what we're classifying as working. So there are some far more challenging ones. So particularly the arenavirus antigens seem tough because they are just hyperglycosylated. They don't really seem to present many classical neutralizing antibody epitopes. They're a bit of a tricky one for all of vaccinology, and the clamp is no exception, unfortunately.
Mhm.
But yeah, I'm trying to think that hasn't worked. I think, yeah, no, I don't think I can really think of it. Because if there are plenty that don't work initially and they require a lot of sort of iteration, as in to sort of get the attachment placed just right. Like we do make quite large panels of variants still. It's not like it's just, you know, put a clamp on and it works. But yeah, usually if you put enough work in, you can find something that is an improvement.
Yeah. I have one quick question, and also with Belinda, in particular with what you have presented. Belinda, question there for you. So you've had your IP chat discussion first, and you have now seen the presentation from Andrew. So do you say that all ticks the boxes for the data disclosure, or what would you say there?
Uh-oh.
Yeah, so there's quite an extensive IP portfolio behind what Andrew's talking about. We have foundation patterns going back till 2018, and then another suite in 2021, and then another suite that have come through this year. And Andrew's actually done all of the right things in that he has flagged that he was presenting today with our commercialization associates. It's all been approved. So nothing that Andrew's disclosed today was actually, it's already in the public domain, so he can disclose it as much as, well, within reason. There's obviously stuff that's still going on that Andrew couldn't talk about today, And that's, you know, that's part and parcel of the process, but everything else is clear to go.
Excellent. Belinda, I got a question here in the Q&A session that starts career-wise. After training as a patent attorney, are there still opportunities to return back to the lab bench, to lab research?
So it's interesting because, I mean, I've been in this field for 15 years now, and I think it was a question I used to get a lot more when I was younger. It's all about currency of knowledge. So I think if you left the lab, for anyone that leaves the lab after 15 years, that's a hard place to go back to because you're off the tools. Whereas if it was 2 years or 3 years or 5 years, then perhaps there would be areas that I could go back into, probably maybe starting at a lower point, because, yeah, it's the tools. And so my science knowledge is still really good because being a patent attorney, you have to be up to date with things. And also I'm seeing things before they are in the public domain. So I'm sort of in on the secrets of what's actually emerging. But yeah, you'd have to, I don't know, I think most patent attorneys quite like their career choice. I don't know if they'd want to go back to the lab. I certainly don't.
Now, Andrew, on that front, I presume you're quite busy in the lab, especially, you know, with Sanofi having shown all this interest as well. What are the next steps for you as far as you can disclose from the work? What's next?
That's an interesting question because it's not actually totally clear what the Sanofi acquisition is going to mean for, I guess, the small sort of group within UQ. So we understand that we want to make more vaccines to more targets, basically. And that's kind of the goal. And so if that is what happens, then yeah, that's what I enjoy doing. So yeah, it's kind of like there might be a bit of flux, a bit of change that's not totally clear at this point, but yeah, I'm hoping just to continue to make more vaccines to more targets.
No, that's excellent. Now, I'm already over the time. It's 7 p.m. here in Brisbane. I would really like to thank you both for speaking, especially in the evening after hours for you. So thank you so much, Belinda and Andrew, for joining the ISV Next Generation webinar here tonight. And with that, thank you and have a good evening.
Thanks, Sandra. Bye.
Thanks, Sandra.
Thank you, Andrew. Thanks, Belinda. Bye.